Um, all right, and then the format for today, it's going to be a little bit of a we're going to do 20 to 30 minutes of an overview from Samantha, and then look through some of the process that she's using in her lab, and then we'll open it up to Q&A. So feel free to add your questions into the chat, and then or feel free to raise your hand, and we can open up the floor to you. So again, thank you everyone for attending, and yeah, making time for us this morning. All right, Samantha, the floor is all yours.
All right. Well, thank you very much, Eric, for the introduction. Like you said, I've been kind of not working for, but working in conjunction with Flexergy for a few years to kind of get their platform, in the veterinary clinics and comparing it to the gold standard, which in our lab is quantitative PCR. So today I'm going to really talk about how to conduct good PCR within your facility, right? So now you guys have this box, which is amazing. It's easy to use, but what about contamination? What about training on it? And also, what's a few things that you can do to keep reducibility, in your facility or your clinic? All right. So we're going to talk about separation of your activities. I like to call this kind of like the clean area and the dirty area in your clinic. Proper ways to sample. You guys are probably very familiar with the analyzer and results. I just wanted to touch bases on that. Training in your lab, and then also checking for cleanliness in your lab. What about lab contamination? Are you getting false positives because your countertop has salmonella on it? So this is something we're going to talk about. So I really highly suggest separating your Flexergy unit, your analyzer, into a clean area within your clinic. And then where you're processing or growing or culturing your feces into a second area within your clinic. I call these your clean area and your dirty area. And this is going to be the easiest way to prevent contamination within your clinic. Ideally, these would be in two separate rooms. And I know logistically that's not possible most of the time. So I would recommend two areas, two corners within a room. It's just as far apart as you can put them, the better it is for your clinic. Also in your clean area, you're going to have your analyzer, all of your reagents, all of your plasticware. Anything that's related to the PCR is clean, right? We don't want any contamination there. Versus the opposite, right? All your sample prep and your incubators is in your dirty area. Also, ideally, when you're going between the two areas, you're going to change gloves and lab coats. At least change your gloves after you've done a sample prep, and now you're going to go test some samples. You kind of can't change gloves and lab coats too often. Now, because of PCR, most of all the samples need to be in a liquid format before we can extract them or test them. So you're going to want to use the buffers that come in your kits, okay? Because not all buffers are created equally. There's different salt concentrations in there. And Flexergy has worked out what buffer concentrations work for their unit and don't give you negatives or an error message. So always use the buffers that come in the kits that are provided. You're going to prep your sample, your feces, in your dirty area, like we've already talked about that. Make sure everything's labeled, right? Particularly you have your fecal sample, you have your sponge bag, and you have your selenite broth tube. So you want to make sure that those all have your animal's name on it. After you've put feces onto the sponge or into the selenite broth, you want to, check your gloves, check your countertop. Especially if the fecal material is very dry or has shavings in it, it's really kind of easy to make a mess. So definitely make sure you wipe down your countertop if you see any contamination. Also, when I'm training somebody new in the lab, I tell them to think that every sample is positive. That way you're a little more concerned, if you will, about getting any feces on your gloves or your countertop. So when you open up that, screw cap, a fecal sample, you want to think that there's salmonella covered all over in there. So you're going to be careful where you set it down. You're not, you're going to be careful setting the cap upside down so you don't contaminate your surface. So just think of everything as positive and that you want to just keep wiping things down. I mean, very vigilant about contamination. You will gain more confidence in your testing ability over time, particularly when you've prepped a really strong sample and then your next sample is negative and you did not contaminate it. You'll gain more confidence over time that you are doing things correctly. Along those lines, only open and process one sample at a time, kind of just normal, good laboratory practice. All right, now, so you have your analyzer, it's in the clean area. You've incubated your biological materials overnight in a separate area. So now you're ready to test your sample. So you're gonna, take your selenite tube out of the incubator, close the cap on it since you've loosened it for a little gas exchange. I like to mix it pretty well. You're gonna take your 14 microliters out, mix that with your master mix, and you're gonna run that. Now, do be careful when you're mixing that selenite that quite often, since it's liquid and it's a screw cap, you do get liquid on the outside. So you wanna make sure you decontaminate it if that's happened. I'm not going to go much into the use of the analyzer because you guys get thoroughly trained on that by Flexergy. I would say though, if you're testing a sample and you do happen to get an error message, I would suggest diluting your initial sample, your initial 14 microliters, 1-to-1 with some PBS. So take 14 microliters of your selenite broth and add 14 microliters of some PBS. Mix that up and then take another new 14 microliters and test that on their instrument. Feces is well known to have lots of PCR inhibitors, and the best way to combat that is dilution. Don't worry about diluting out your salmonella. I mean, obviously that's a possibility, but I've done curves where I dilute a positive broth, 1-to-1, 1-to-2, 1-to-5, 1-to-10, that type of thing, a nice serial dilution. And you can take it pretty far and still get a positive result. And just a little bit dilution will go a long way. I'm going to stress here to not take any shortcuts with PCR. PCR is one of those techniques that you can't change very much. All right, Flexergy has already taken all of the shortcuts into account and done them all. So don't change a thing. Once you have a protocol that's working in your clinic, don't change it. This will give you much more confidence in the reproducible results, and also it'll be easier to train people. And talking about training people, I think I would really start with just 2 or 3 people within your clinic trained on this technique, using everything, and being very aware and observant of what's going on. Get them highly trained and then start adding new people that can run the testing for you. One of the things I noticed the most that's important for qPCR is your observational skills, okay? So when you're adding the 14 microliters and you're taking your 130 onto your test card, there's always a little bit of volume left in that 1.5 mil tube, right? So on one day, does it look, let's say average, but then the next day, you've got like 3 times as much volume in there. Well, use your observational skills, acknowledge that something went wrong, right? Why is there extra volume in that tube? Was my pipetter not set correctly? Did I have an air bubble? Did some of the fecal fibers kind of stop liquid from being pulled up in the pipette tip? Any of that stuff is where you want to stop what you're doing and start over, Okay, so use your skills, go slowly and become very familiar with the process and don't change anything. One of the other main things about running PCR on your lab that a lot of people become concerned about is cleanliness, okay, contamination. So not only may you be testing for salmonella in your facility, you may be growing it too in the incubator. So now you have salmonella that you know is growing. So the best way for PCR, the best way is to physically scrub an area is the best way to remove any contamination. Okay, you can use 70% ethanol on a paper towel, or you can use sanitation wipes. I just want you to really think about scrubbing the area. Remember, PCR can detect live and dead viruses and bacteria. And also because the DNA is so small that we're detecting with these units, you have to really chop up the DNA really finely before the PCR to be negative. So you need to make sure that you physically remove the bacteria from a surface. And you're going to want to clean each area before and after you use it. So what I would suggest and what we do within our own facility is every other week, we test our clean area in the lab and we test our dirty area in the lab. So what I would recommend you doing is just taking one sponge, and I'm going to have a video later on about this, and just swabbing or sponging that whole area. You're going to do your pipetters, you're going to do your countertop, anything that you may have touched with your dirty hands or you think salmonella may be growing is what you're going to test. And you're going to do the same thing with the new sponge in the dirty areas. So you're going to take out your 1 ml, put it in your selenite, grow it overnight, and then you're going to test it the following morning. So not only can you check the cleanliness of your clinic or your lab that you're using, you can do the same thing in the environment of your whole clinic. So you can test, stalls, barn drains, buckets, sinks, barn doors, equipment, the staff offices, telephones, medication areas, anywhere where salmonella could have been, traipsed through your clinic. You can use your sponge that area really well, incubate it and test it the following morning. So my take-home messages here before I show you the videos is to separate your activities, right? Separate your clean area and your dirty area. This will give you so much more confidence in not worrying about false positives in your clinic. Clean, clean, clean. Test, test, test. These are kind of obvious. It's not like it's taken a lot of time for you to test these, that you don't have lab contamination. Twice a week, 2 tests. That's pretty minimal activity that you need to do for that. And please don't take any shortcuts and do training. I know this is a really easy platform to use. I just don't want you to start taking shortcuts. So I have a couple of videos here. I'm hoping they're going to play okay. And here's one of the first ones I'm going to talk about. So what if you wanted to test feces directly? Okay, what if you did not want to go through that selenite incubation? What if you had a sick animal in your clinic and you wanted to test them right now and you wanted to know within the hour if they had salmonella or not? Okay, so I'm going to talk about how to do this with a FlexerG unit. The most important part here is how much feces you're adding. Okay, like I said, feces has a lot of PCR inhibitors. So actually smaller amount is better. And you want your feces and PBS mixture to kind of look like weak tea. Okay, it should be almost a little transparent. It definitely should not be mud or dirt color. So let's see if we can get this to play.
In this video segment, I'm going to be showing you how much feces to add to 5 mL of PBS if you want to test feces directly and not go through the selenite enrichment. Now you might want to do this if you don't want to wait an extra 24 hours for a result, but don't forget, the enrichment is going to increase the bacterial to grow, and so this might give you a negative result. Now, as you know, feces has inhibitors. So you can definitely add too much feces to this mixture. So I have 3 different sizes of fecal material here. We'll call it about the size of a grain of rice, peanut size, and maybe an almond size, okay? Now I'm gonna add these to PBS, mix it up, and you can see the color of this liquid afterwards. We want the color of about a weak tea, okay? So definitely less is more. So that's kind of a nice looking color. Let's see if this medium one, how that looks in here. Come on, you last little bit. There we go. This is pretty dark color. And then the last one, which is going to be way too much, is going to be looking like mud in here. But I want you to see the differences. Oops, spilled some on the counter. Talked about contamination earlier. Well, we will clean that up. So, pretty much the color of mud. So you can see sample 1 is definitely a nice weak tea color. And this is the color you want to be at if you're going to test fecal material directly on the Flexer-G unit. Now, we've talked about having your dirty area and cleaning up contamination. So I'm going to grab my sanitation wipe here. Clean this up. While I'm here, I'm just going to put these all together. Dispose of these in my medical safety waste and decontaminate my whole area. Now you're ready to test these on the Flexer-G unit if you wish to do so.
Alrighty, so really easy to do. All right, the second part we have here is what about using your sponge and also doing your selenite enrichment? So if you did not want to test your direct feces, I've kind of come up with this modified version of using your sponge and your selenite to test different, to test the fecal material. So I'm just going to play this video here where I show you kind of how I've come up with this new technique.
So this video segment, I'm going to talk about how to sponge a fecal sample, incubate it into selenite, and then test it the following morning. Now I do feel a little silly showing you veterinarians multiple different types of fecal sample types. So we have your dry feces, which you guys, fully aware of. We have a normal fecal material. We have some feces that is loaded with hay. It could also be one that had a lot of straw in there. It's particularly diarrhea that fell on straw for a foal. We have reflux, very liquidy form. And then we have a rectal swab if you guys choose to go that way. So what I'm trying to demonstrate here is the flexibility and the dynamics of the Flexer-G unit. All 5 of these will give you ample fecal material to incubate in your selenite and give you a result the following day. So when I have a sample like this and I want to test it the next day after enriching it, I'm going to cap it up, shake it all around, and I'm going to sponge just the inner face of the lid. Because like what we did in the previous video, you want just a little fecal material. You don't want a lot. So I'm just going to sponge this lid. As you can see, you're getting just some on here, not a lot. Put it in my bag. Cap up my sample. Mix it around just like you would normally do when you're testing for an environmental sample with the Flexer-G platform. Get your nice brown juice here. You're going to take out 1 ml just like you normally do. Nothing's different here. All these volumes are just the same. Then you squeeze some out. Take my 1 ml. I have my selenite here labeled. I always like to shake this up really good to get everything mixed. Particularly you can see there's a little sediment in the bottom. Get it nice and mixed here. I'm going to loosen this cap before I go into incubation so some of the gases can off-gas if they need to. Now I'm not going to show you the rest of these because that would be a little silly. I will say though, on your rectal swab, if you do choose to go that route, just put the whole swab in here. It doesn't matter if you have the applicator on the top part or the bottom. You just want to make sure that the part that has the fecal material is actually in the liquid. Okay, so if you had a really long swab, you might want to break it off because you want this contact between the fecal material and the selenite so you can have salmonella growth if that's what we're looking for. Again, I'm just going to give it the shake. You can't hurt anything at this point. All right, again, we're going to twist that top off a little bit for off-gassing. We're going to place this here. After I would have processed the rest of these, this would go in the incubator for overnight, and then we would test these the following morning. So this is a good example of the dynamic range of the fecal material, especially the different consistencies that you guys normally see in your practice, and how well this will work testing for salmonella directly with your selenite broth.
All right. So I'm really happy to see that this product can be used so diversely, especially with all the different fecal forms that we get in the lab all the time, especially like the reflux or shavings where there's not much material in there. There's still enough in there for the Flexergy unit to detect selenite. Now, my last one here, we talked briefly about testing your lab for contamination. I would recommend doing it every other week, just, write it on the calendar. And it just becomes a part of your normal practice. So this is just a quick little video showing you how I would sponge the clean area within my lab.
All right, in our last final video segment here, I'm going to show you how to test for contamination within your own lab. So this is my clean area in the lab, which we have spoken about during the talk. So I'm going to take my sponge and I'm going to just rub my sponge across the surface. You can see you're kind of leaving a nice little wet trail anywhere where I may have contamination. Your pipetters, particularly the little top part where your thumb and fingers are going to go, the bottom part. All this is where you're going to test for contamination. Put this in your bag, mix it up, and then you're going to take out 1 ml, just like you normally do, incubate it in your selenite broth overnight, and then you're going to test it the following morning to see if you have any growth from salmonella. You should always have a negative result, so let's keep our fingers crossed. But this is just a quick, easy way to make sure that you are not accidentally contaminating your samples and that you have a nice, clean work environment.
All right, so contamination, right? The only way you're going to know if you have it in your facility is for you to test for it. And it's a great thing to do because then you have confidence that you can tell everyone, your clients, your coworkers, that your area is free of these pathogens that you're testing for. And I think that's a good feeling and a good thing to have within a clinic. So that's the end of my presentation. I just hope I touched upon a whole bunch of different little areas where you can think about how you would implement this within your clinic. If you're already doing this, if you want to start doing that. So I think I'm just going to send it back over to Eric and we'll see if we have any comments or if there's any questions that people have.
All right. Yeah. Thanks for that, Samantha.
Sure.
Always learning. I think, again, to everyone, if you do have questions, please put them in the chat, or again, feel free to raise your hand. We can go ahead and ask questions as they come in. Let's see. Oh, I'm getting something that the chat is disabled. Let's see. Hmm. All right. Well, let's see. I'll if you do have a question, you could feel free to just go ahead and introduce yourself and ask it. But I have a few questions on my end because I think it's always interesting. You know, we definitely made the pop or the test for environmental surveillance, and you know, I think a lot of clinics want to use it for more clinical testing. So we appreciate you creating a different workflow and improvising there and validating that. In terms of UC Davis, because I think, again, this is a new type of technology. Clinics are maybe not used to using this technology or PCR testing in their own labs. You know, what does UC Davis do in terms of like the surveillance program at the veterinary teaching hospital? It's really interesting to understand the frequency and kind of the process that you guys are using.
Yeah, so the actual veterinary clinic where the horses are being, stalled or housed, they get tested. They have, I'm going to say, about 50 different sites within all the barns, the isolation, NICU. So there's a total of 50 sites that they test once a week for selenite. We do a slightly different process in that we pool, so we test each site individually. We get our selenite broth from each one of those sites, and then we combined 10 samples and do one single test. So we pool 10 different locations and do one PCR test. And if that's positive, then we test each of those sites individually. It's just a way to save money on the PCR side of things. Within our own lab, our PCR lab, we test every other week, just like I'm recommending here, but we test for every pathogen that's been positive during that 2-week process. So we might have, equine herpes virus 1, equine herpes virus 4, streptococcus, equi, canine herpes virus, who the heck knows? So we swab, we have 12 different areas in our lab between the 3 different rooms that we swab, and then we test for all of the pathogens that were positive. The area that's usually positive is our large centrifuge where we're spinning down the raw sample. Mm-hmm. So that's often, I'd say once a month, the centrifuge is positive. And so we just give it a really good decontamination, scrubbing, hot water, bleach, and all that goodness. And then we retest it again because you always want to make sure you cleaned it up. So that's our process.
How many, I guess when you pool, right, I think for us, I don't know if our system is validated. We did pooling during COVID actually, but how many samples do you pool typically when it comes to the PCR testing side?
Only up to 10. I've done a validation study up to 10 samples, and we don't dilute out a weak positive. Up to 10, we can still detect the weak positive. But after that, we started diluting it out. So 10 and under, I feel comfortable pooling.
Okay, great. Yeah, I think, and that's like actually I have a follow-up question there where, so you mentioned you validated the pooling protocol. So what does that mean? So when I think it's always when you're trying something new, like you mentioned, or if there's a product, you should follow the protocol. But when you're trying something new, you have to validate it in a way, right? So, what does that mean in terms of validating? And, let's say you get a new technology, it doesn't even have to be our platform or whatever it is, right? Like what does that validation process typically look like from your perspective?
So it's always a challenge to bring new equipment into the lab, or especially new technology, because you're excited and scared at the same time, right? So we usually do a pretty lengthy, about a month validation process where we're doing side by side with the gold standard, or what we're currently doing, and with the new product. So if you are currently, sampling your fecal material and let's say sending it to IDEX or sending it to whomever, you're not doing it within your facility. And now you have this Fluxergy analyzer in your lab. I would probably for at least a couple of weeks be sending half my sample to my normal outside lab and then doing it in my clinic myself. And that'll give you confidence in your positive and your negative results, right? Hopefully during that period of time, you do have a couple positives and hopefully lots of negatives, but that way, you just gain confidence in that you're detecting it. Let's say you go through a 2 or 4 week period and all your salmonella samples are negative. So you're like, okay, let's do it in our lab now. We're feeling good. And I would just do that. And once you get a positive in your lab, like you might want to go ahead and send that one off just to make sure that, another place got a positive result also. So yeah, take some time, right? You want to have a lot of confidence in your results. Eric, I don't know if you can see it, but I can see there's a couple Q&A highlights. I don't know.
Yes, yes, there are. So, yeah, I think this is a question here about the selenite tubes. So,
Yes.
Samantha, any suggestions for technicians to mark their selenite tubes as loosely versus firmly capped? With the inverting and mixing going on, have you determined a clear way for teams to easily know if the tubes are open?
Good question. So for us, our tubes are the only time our tubes are open is when they're in the incubator. So if you're getting a whole bunch of samples ready, let's say throughout your day, and your tubes are sitting out on the countertop, they're all going to be closed tightly. Okay. So right before they go in the incubator, whoever job that is, that technician is going to go through and loosen them all, put them in the incubator. And then whoever's job it is in the morning to take the selenite out of the incubator, their first action after they close that incubator door is to screw on all the caps. Okay, it's also a good way to prevent contamination, right? Because if your caps are open all day long, you can have bacteria going all over the place. I know that's not how it really works, but that's how you need to think. Okay, if that cap is left loosely open on the counter, is salmonella going all over the place? Probably not, but you don't want to take that chance, right? So you want to keep it closed. So that's how we do it.
Awesome. No, yeah, I think I can resonate with that as well. And I think with tubes, also labeling. Is there a certain protocol you guys have in the lab when you guys are, you're dealing with hundreds of samples, if not more, per day? But what do you stress with labeling samples, especially when you have, 5, 6 people working in your lab?
Yeah, just every sample has the patient's first and last name. We have not moved to a barcoded system yet. We're on our way on that. But right now, every tube just gets a handwritten because your selenite tube is, not the original container that the feces came in. So every selenite tube just has a handwritten, Holly Barnum on the side of the tube. So you know which sample that is. So that's just how we do that.
Okay, great. Another question coming in here. So going back to pooling, what volumes do you typically pool? I guess it really depends on what you're pooling, but yeah, would love to hear your perspective.
It's actually not very much. So if we have like those selenite tubes that come with your kits, which I think are 12 mils, I think 12 to 15. So I would, if we were going to pool 10 of those and do one PCR test, I'd probably take about 50 to 100 microliters out of each one of those selenite tubes and put that into a 1.5 mil snap cap tube or just another container and just give it a good vortex. Now save those original selenite tubes, because if your pool is positive, you want to go back to your original sample and do your retesting and find out who was positive, right? But yeah, not very much liquid. It's, I'd say, 50 to 100 microliters. You could do more. It's not going to hurt anything, but you don't have to do very much.
Okay, no, yeah, that's great. And I think there are also some complexities to pooling, right? And you have to change your pipette tips, and I think just like anything, like you mentioned, it seems like that validation process, giving yourself the time to try it out and compare it against, let's say, UC Davis, the gold standard for this type of testing. So yeah, I think that's definitely a good insight there. But yeah, you want to practice, especially if you haven't done it before. It's kind of like baking, but a little.
Yeah, I think you can play around, use those words loosely, with sample prep and pooling and that stuff. I think that part you can play around with that, but I don't want you to play around with the actual testing, right? The volumes that FlexRG has, the storage conditions. Don't play around with that part. You can play around a little bit with sample prep, but not with the PCR process. That part cannot be changed.
Okay, great. All right, lots of good questions coming in. How about, the incubator? So the incubator is, you're now culturing stuff in your clinic that you haven't cultured before. Do you have a standard cleaning process? I think going back to the environmental, routine kind of QC surveillance, is there anything that happens there with the incubator in terms of testing side and then also cleaning?
The part that gets cleaned the most, to be honest, is what we hold the tubes in. So that tube rack, often stuff bubble up over the sides, especially we don't use these specific tubes in our labs. We use something else. And sometimes they bubble over a little bit if you get them a little too full. So your tube rack, we get a lot of contamination on that, but the actual incubator, we don't. It smells nasty, don't get me wrong. But we rarely get contamination in there. But I would just use your sanitation wipes or your ethanol, spray it down, wipe it with bleach, wipe it with ethanol type of thing if you need to clean it. Often we will also put a layer of tin foil on the bottom and set our tube of racks on that. So if we do get overflow or we think we might have contamination, we can just take that tin foil, kind of like what you said, baking, take it out of your oven and throw it away, and then you don't have to clean very much, right? So sometimes it's often lined in tin foil to help aid the cleaning process.
Awesome. Yeah, no, I think it seems like process is really important here. Just making sure you have these routine processes that you can follow to keep it easy.
And that's why I also like only a few people doing this, right? Because then that process is easy and only a couple people know how to do it, and they're always doing it the same way. The more people that are jumping in here, they're going to tweak the process to what they like, which is normal human reaction to things. But you don't really want that with PCR. You want the process to be uniform, and you want your testing to be uniform, and you don't want people to tweak it. You want it to be the same every single time.
Yeah, perfect. I also want to, I think maybe with just general questions of PCR, it's always interesting to understand, the background behind what you're detecting and knowing like what assay you're using, and, even going, maybe the gene level is too much information, but it does give you information when you do understand it. Can you go a little bit into how UC Davis develops their assays, or how you develop all of the assays at UC Davis, and kind of the work that goes into that? 'Cause I think it's really interesting to understand, it's not just a black box, what's behind it.
Yeah, there's a lot that's involved in to bring in a new pathogen, into fruition for our lab. First of all, it's the need of it. Are we getting a request for this once a year? It's probably going to not make me any money, right? Am I getting requests from this, every week? Okay, veterinarians are really interested in this pathogen. Let's take a little bit, of a better look into it. So the first place I always start is literature. I do a literature search, right? What are the experts in this field? What genes are they looking at? Is an assay already designed for this pathogen? Can I just tweak the assay a little bit to work with my instrument? That's ideal. And so I just do literature searches. The main thing to also really concentrate on is mutations or different variants of the different strains, right? Salmonella, you have all these different isolates, but we want one test to pick up many, right? So we need to look for a conserved region within all these different strains of Salmonella that I can design one single test. You don't want to have to test 10 different PCR assays to say your animal doesn't have salmonella. That's not realistic. And so we look at all the different strains, find that homologous region where I can design one single PCR test. And then we just start testing samples. We also order, if we can, like a pure culture, from a institute that we use as our positive control. Sometimes we make synthetic or fake positive controls where it's really just the DNA. And that allows me, the good part of getting synthetic DNA in your lab is that you can do an absolute quantification. So I can say like, hey, this assay is so good, it's detecting 10 copies of this gene. Or this assay is so bad, I'm only detecting 100 copies. This assay is no good. Go back to the start. So we just test lots of samples. We run it against all different sample types. That's not really so much a problem when you're designing a new assay. But yeah, I just do lots of ground research before we start on anything to see what the experts are doing out there and can I copy it, essentially. Why reinvent the wheel? If I can just take someone's assay, modify it so it works for me, then I'm much happier.
Oh yeah, thanks for sharing. Yeah, okay, lots of questions here. So let's see, does UC Davis send positive samples out for serover testing?
We do not our lab. We do it when the clinician requests it. So sometimes we get clinicians where they get a positive result from us, and then we send the sample actually to the teaching hospital where they grow it up and do the culture and sensitivity of it. But we don't do any of that in our actual facility.
Mm-hmm. Okay, great. And I mean, maybe I don't know, you can discuss quickly on some of the all the assays that you have available, or just because I think you do have, let's say if you do have our platform in the clinic, I still always like to, always have your central laboratory resource. I think they work hand in hand versus one versus the other.
Right.
Maybe you can quickly discuss, some of the services that you have at UC Davis and you've been providing for the whole industry.
So our lab, we're not actually part of the teaching hospital. We're a separate core facility on campus. And we probably have close to like 150 different pathogens that we offer PCR testing for. And that's for dogs, cats, horses, llamas, pretty much any species, especially like salmonella. Sometimes we get iguanas from zoos, or we get exotic animals or rhino poop to see if the rhino has salmonella, which is really one of the really nice things about some of these pathogens that are not species specific, like not like HV1, where you're not going to see that in a dog, but your salmonella, your Clostridium difficile, your Lawsonia intracellularis, any of those, right, you can test multiple species on there, which is a great thing about PCR. Sure, I can test a dog for equine herpes virus 1, but I'd be pretty much wasting your money, right? That would be patient 0. We haven't seen it before. So probably not a good choice of cost there. But yeah, so we offer lots of tests. Our lab only does qPCR. So we only culture salmonella through selenite for this enrichment. So we don't grow anything else in the lab. We don't do ELISAs. We don't do IFATs. All of those other techniques, the teaching hospital has, facilities for that. We're just the specialist in just qPCR, and that's where we're staying. And I think that's what our lab is all known for, is quick turnaround time, next day turnaround time, and being able to process, hundreds of samples a week, just to get the results out there as fast as possible.
Oh, great. I think, it's definitely invaluable for the whole community, and a lot of samples are coming through your lab, so
especially outbreaks. Those are the rough times when there's hundreds of samples at one time.
Yeah, we don't wish that on anybody.
No, absolutely not.
Let's see. I do have a question. It's more about like the reporting. So, now that you're so you're a PCR lab, and essentially, so right now we have salmonella, but in the future, we plan to have respiratory disease tests and things like that. How does that work with, vets or even your lab with reporting, right? Because it is a responsibility now that are essentially acting as the lab. So
Right.
just curious whether you're using our system or, again, you set up a larger system. Now that you're conducting the test and getting the result, how does that look with your group and even what you tell clinicians?
Yeah, so in California, which I know you're probably not all from California, the CDFA, the California Food Animal Health, has a fact sheet, and it says that anyone that's aware of a certain list of pathogens, that you are supposed to report it to your state vet. And so since we get samples from all 50 states, I just literally go off of which each state has put into law or is their regulation. So you're going to need to check that. Unfortunately, every state is different, which makes it confusing when you're being the one that's reporting it. So I'll talk to California because I'm the most familiar with that one. So once a month, I send a report to our state vet giving a list of all the positive Streptococcus equi, EHV1, EHV4, and influenza samples that we have tested positive in the lab. And so I just put it in an Excel sheet. I put, the only information they get is the veterinarian's name and the clinic and what the sample was positive for. There's no animal information that they request from me. Like I said, I send that once a month, and then I don't know what they do with that information. I don't know how they act on it, type of thing. So there's other states where EHV4 or Streptococcus is not a reportable disease, so you don't have to do anything about it. I'll have to say the default that I always go by is EHV1, particularly if the horse is neurotropic or is having neurological signs, and influenza. Those two pathogens every state vet wants to know about. And again, I only do those once a month. I do EHV1 neurotropic. I do report that as soon as I'm aware of that result, I do report that to the state vet because often quarantine has to be implemented and people are really concerned about that disease. So I do let most of the state vets know immediately. But yeah, it's different everywhere. So you're just going to have to be aware of whatever the regulations are within your state. And what's being tested there. But I just have an Excel sheet with all the states' codes on them and which pathogens they want. Some of them want to know about salmonella. I'd say maybe a handful of them do. Most of them don't care. California cares if it's a bovine. They want to know if it's salmonella positive. That doesn't matter if it's equine positive. So look at that too. Each of the lists are divided by the species along with the pathogen that is being detected. So it's all over the place. There's no federal regulation. It's all state.
Sounds like people should talk to their state vet.
Yeah, it's just a lot, but hopefully you have this unit in your clinic. You're only testing samples from your clinic. You only have to worry about your single state. Maybe if you're back east somewhere where you have the states are all close together and it'd be easy for vets to drive all over the place. That's not such the case here in California. So it might be a little bit of issue back there. You might need to think about which state the sample is coming from. And actually it's, where the horse is, Now as a reference lab, I don't always know where the horse is. I know where the vet clinic is, but that doesn't mean that's where the horse is. So sometimes I know the state vets have to go on a little hunt to find out which state the actual horse is in, because that's what they're really interested in.
Mm-hmm. Okay, no, that's great information. Yeah, I think, it's like lab practice, but also, what do you do before you collect the sample, while you're collecting the sample, when you get the result? I think these are all great pieces of information.
Good.
Maybe one last question. I think this is a more practical one from one of our attendees. So you mentioned lab coats. I think we see you in your videos, you're wearing a lab coat. Maybe you could talk about that. Is that something you'd recommend to vet practices? I haven't seen too many when I go for
Yeah, I mean, We're required by our institution to be wearing a lab coat while we're doing any science, and generally gloves. I would say if I was in a clinical setting, in a clinic, I would probably wear a lab coat when I'm processing the feces, because that's where, especially when it's all hay and flaky and all over the place, um, it gets on the countertop and on your gloves pretty easily. So I think if it were me personally, and you're not, like wearing scrubs, you're wearing your regular clothes, which is what we wear in the labs. We wear our regular clothes and then we wear our lab coat and gloves over that. So I would probably be inclined to wear a lab coat when I'm prepping the sample, like when I'm trying to get it into the selenite tube. I'd have to say after that part, I would probably feel comfortable not wearing a lab coat. When you're taking out your 14 microliters or loading the chip and doing all that part, I would probably still wear gloves because again, you don't know if that sample has salmonella in there growing, right? So I would still be wearing, if you're not going to wear a lab coat, I would definitely wear gloves throughout the whole process. So that would be kind of my personal, like, minimal PPE, if you will. That would be what I would suggest.
Yeah, no, I think that's great. And also like when you put on the lab coat, I feel like it's also, kind of puts you in that mindset, hey, I'm doing lab work right now. I need to be focused and focus on process, cleanliness, things
Right.
like that. Not multitasking, etc.
I agree.
Say,
Yeah, definitely, along the lines of cleanliness. And it just gives you more confidence that you're not spreading things around the lab too. Or from patients. Like if you were wearing a lab coat while you're seeing a horse, which would be kind of silly, but if you were out seeing patients with a lab coat and then brought in all that dirtiness into your clean area, that's not good either. You don't want any of those dust and debris and fecal material and blood that gets dried on your scrubs. I don't want that in the clean area in the lab. Okay, you want that to be clean. That's why I talked about that. So you would want to either change your clothes or put a lab coat. It can be one of those disposable blue lab coats. It doesn't have to be one that has to be laundered, and you can wear it for a couple of weeks, especially if you're only wearing it for like 5-10 minutes while you're prepping samples. It's not like you need a new one every single time. No, not at all. Wear it till it gets dirty or smelly. That just gives you an extra barrier.
great. Okay, sorry, we 2 more minutes here. I will ask you one more question. Samples, right? And you're storing, like, let's say, you incubate 5 samples and you have to store the samples in the fridge, and maybe you also have your raw feces in there and your swabs that you're waiting to send out. Do you recommend keeping like a clean fridge and a dirty fridge, or is that how you guys work in your lab?
A little bit. We have a sample fridge. Okay, so any sample can go in there throughout the whole process. All right, but soon as I have DNA, which is not part of how your process works, that does go into a clean area. So I think as long as I think it's totally fine for you to have one refrigerator that has, your raw feces, your feces that's in your selenite broth, and samples being ready to be sent out. That's totally fine to have that all within one refrigerator. We put them in buckets, right? So we kind of have like our raw feces, Tupperware tote, our tube of selenite tubes that are in a tote, so they're all standing vertical, and then another rack of all of our new samples that have just come in. So we kind of keep it all separated. So it's kind of like feces, if they're in that Tupperware, that they've been done. Selenite's kind of obvious because it's in a tube. And when they're in this rack over here that's labeled, like new samples, those need to be processed. So they all go in the same location, but they just have different bins that we put them in depending on where they are in the process of being run. And we normally store them, raw feces, we normally store in the refrigerator for like about a week or so. The selenite, probably about the same. And just kind of like every Monday, we just go through and throw the previous weeks away. Sometimes we keep them a little longer. Usually it just depends on storage and how much room we have in our refrigerator to store all the quote old samples. The only time we ever really need to go back to an old sample is if it is salmonella positive and the clinician requests, culture and sensitivity, then we need to forward that sample to another lab. But other than that, we rarely have to go back to that sample because you're going to get your quality, right? As soon as you get a negative result or a positive result on your analyzer, you really don't need that sample anymore. You shouldn't be questioning it, so you shouldn't have to go back to incubate it. So you can probably, I'd say within a couple of days, throw away your samples.
Awesome. No, I think that's great information. And again, it's like, process and making sure, the more like little steps that you put in, it'll make it easier to, or have less mistakes. But
Right.
That hour went by quickly. So
Good.
Samantha, thank you so much, for your continued contributions on the research side and always kind of pushing, the testing and industry forward. And thanks for always helping us out and
Sure, anytime.
Contributing to research there. So thanks. Really appreciate it.
You're welcome.
All right. We can drop Samantha's information to anyone that's attended if there's ever any questions or anything like that. I think she's a great resource. Thank you everyone for spending your morning with us or afternoon, depending on where you're calling in from. Thanks again. Have a great day.
All right, thank you. Bye.
Thanks.